Hey, my name is Brian Derby, currently Jim Grotting's fellow. I really appreciate the ASJ editorial committee selecting this as the best research paper in ASJ for 2014. The majority of this work was done while I was a resident plastic surgery at SIU in Springfield, Illinois. The title of my article was Adipose-Derived Stem Cell to Epithelial Stem Cell Transdifferentiation, a mechanism to potentially improve understanding of fat grafting's impact on skin rejuvenation. It's a long title, but I'll get to the details so that the audience fully understands. We have no financial disclosures. So really what I hope everybody that's listening today walks away with from this talk is that we'll clarify what we currently know about that so-called fat stem cell effect on overlying skin after a fat graft is placed. But with this talk, I hope to offer another possible explanation for this effect that has not been previously explored. And in order to do so, I'm going to introduce the concept of P63 to the plastic surgery audience, which is an epithelial stem cell marker. So a little bit of background, a basic PubMed clinical query on ADSCs will show you that it's an extremely popular and hot research topic. There's over 500 studies dedicated to these cells on their therapeutic application. There's over 30 systematic reviews and over 1,000 studies just on the medical genetics of these cells. How we bridge the gap from cell biology to plastic surgery is through fat grafting. Oftentimes after fat grafting, we all know that there's an improvement in skin texture and appearance overlying the grafted fat, but how exactly does that occur? Well, the labs in California with Dr. Longacre and in Italy with Dr. Rigatti have shown us in detailed fashion that there's an improved blood supply and an ultrastructural change to the dermis that occurs overlying grafted fat. We feel that ADSCs are likely contributing, and these previous authors have shown us that this is likely through a paracrine mechanism, or basically a payload of proteins being deposited into the skin or influencing the overlying skin after the cells are placed. But this got me thinking, perhaps there's another way that the cells are contributing to skin improvement. And therein lies mechanisms of cellular differentiation. We know that ADSCs can differentiate into keratinocytes within the epithelium, but a keratinocyte is an end product of cellular differentiation. The thought was, for this manuscript, are ADSCs capable of reprogramming themselves or transdifferentiating from one stem cell type to the other? So, in order to get into the details of investigating that question, first you have to come up with the concept of how do we identify epithelial stem cells? Well, a literature review will show you that P63 is indeed an epithelial stem cell marker. It maintains epithelial cell proliferative capacity and cell layering. If you look at the image to the immediate right, you can see that without P63 and with P63, there's a stark difference in the epithelial thickness that's shown in a previous mouse model. Its expression is also high in progenitor cell layers of skin, prostate, and the breast. So our aim was basically to use labeled ADSCs to track their progress after we engrafted them into a mouse model, and then to determine if they're capable of P63 expression, that epithelial stem cell marker. So our experimental design, like I said, we tracked the stem cells that we placed. How do you do that? Well, we used a green fluorescent protein labeled mouse, all the cells of which fluoresced green. We then placed their fat into a nude mouse model to limit the immunologic reaction to the cells after placement. And we put the fat in one of two ways, either as whole fat or in group 2 as isolated and purified ADSCs in a peptide hydrogel. A peptide hydrogel is basically a construct that carries the cells such that when you inject them underneath the skin of the nude mouse, they won't disperse freely in that deep to the subcutaneous plane. So our methods. As I mentioned in group 2, you have to isolate and purify the stem cells before you put them into the nude mouse. So after fat graft harvest from the GFP labeled mouse, you subject the cells after breaking them up to fluorescent activated cell sorting, which is a fancy word for basically plucking the cells of interest from that heterogeneous mixture of cells that came from the donor mouse fat. We plucked the cells out of that sample that were positive for CD34, 73, and 105. And if you do a literature review, you'll find that those 3 cell surface markers typically identify ADSCs. So as a visual example of that process, these are our GFP positive cells here. They're blue nuclei, and the cell surface marker of interest, CD73, glowing red. When you merge these images, you get this top center image which shows you orange, which is the green and red combined. That identifies the fact that our ADSCs were positive for CD73, as I mentioned before, when we isolated the cells. We did this also for 105 and 34. And then to make sure that our cell mixture was pure prior to injection into the mice, we made sure that the sample was negative for these other cell surface markers such as endothelial cells, white blood cells, differentiated epithelial cells, and most importantly, we made sure that our sample was negative for P63, the epithelial stem cell marker we wanted to see over time develop. We made sure that our sample was negative before implantation into the nude mouse. So we took the nude mouse, divided their backs into one of two particular situations, the left side being the experimental side of the mouse and the right side being the control. We elevated the skin flaps and then placed fat inside of those, underneath those skin flaps in one of two ways, the whole fat or ADSC plus peptide hydrogel group. The right side was a control and it was subjected to either sham surgery, which is basically elevating the skin flap and closing it, injecting saline, or just injecting that peptide hydrogel carrier that was carrying our ADSCs in the experimental mice. So then at 8 weeks, we harvested these skin flaps to give it adequate time for those cells to incorporate, for angiogenesis to occur, and for fibrosis to occur if it was going to. And then we analyzed the skin flaps for their gross morphology, used fluorescent microscopy to identify the P63 epithelial stem cell marker, and we used RT-PCR to compare mRNA expression levels of P63 over time. So what did we find? On gross morphology, there was no surprise here. We already know that fat cells and ADSCs increase VEGF and increase vascular ingrowth to the tissue surrounding the grafted fat. And indeed, we showed that with the whole fat. Here we see blood vessels growing immediately atop the fat graft. That was supported in group 2 as well with the hydrogel plus ADSC group. Here we see at 1 week, and then here we see it at 8 weeks. If you compare the experimental skin flaps to the control flaps, you see a much richer and denser vascular network in the experimental skin flaps than you do in the control skin flaps, which makes sense since the experimental flaps had stem cells nearby. And then moving on to fluorescent microscopy, getting to this concept of migration of cells from our donor fat. This is group 1. This is the whole fat group, and for orientation, the whole fat is on the lower aspect of the image and the skin is above it. I'm going to point your attention to the white circles, which show the GFP positive cells from this whole fat group that have migrated into the overlying skin. But this is whole fat. It's a heterogeneous mixture of cells. So how do we know that those are actual stem cells? We can't be particularly sure yet. So we have to look at group 2, which was just the stem cell injected group. First, we have to confirm that our stem cells are alive and well at 8 weeks when we harvested the skin flaps. So if you look at the low power view on the top right with the hydrogel carrier clearly suspending our green adipose-derived stem cells, and you look at the high-powered view, at 8 weeks we can confirm that our cells are alive and well. So that step confirms, so now we can move forward to analyzing in greater detail and getting to the crux of the study, which is this slide. If you take the left column combined, it'll make this middle image. You take the right column combined, it'll make that middle image. What you see here highlighted around in the white circle is that our cell surface marker of interest, P63, that epithelial stem cell marker, combines with the green GFP positive cells that we implanted there to make this yellow light brown fluorescence that you would expect, indicating that the cells we placed in those mice have moved into the overlying skin and are now expressing P63, which was the interesting find of this study and is suggestive of the fact that ADSCs are potentially capable of becoming epithelial stem cells. And then when you look over time with group 2 of P63 expression at 1, 4, and 8 weeks, you see this temporal increase in P63 expression. Now, whether that's from increased existing stem cell activity in the epithelium or whether that's from the ADSCs populating the epithelium and then replicating, it's unclear yet, but it's still something to note for future studies. And then when you look at the whole fat versus ADSC P63 expression at 8 weeks, you see that both groups has significantly increased P63 expression compared to control skin flaps. So because these two groups are comparable in terms of their P63 expression, it's likely that the ADSCs are responsible for the effects seen in group 1. So in conclusion, what we're able to say is that, indeed, our peptide hydrogel scaffold kept the ADSCs alive and well in 8 weeks, so that was useful for future study. We found that ADSCs may, and I use the word may because I'll explain a little bit more about that in a minute, may migrate into the skin and express P63. And we also saw this temporal increase in P63 expression over time, which is likely secondary to ADSC influence in that whole fat group. So for future study, just to clarify in detail a little bit more what we found here, it would be helpful to co-culture or put two cells together in a petri dish and do DNA microarray to kind of figure out this so-called genetic conversation that's going on between epithelial stem cells and ADSCs, to determine if increased p63 expression equates to more epithelial stem cell replication or activity. I don't know, but it's for future study. We also need to kind of work out those pathways between ADSCs and epithelial stem cell transdifferentiation. ADSCs are a mesoderm-derived cell. Epithelial stem cells are an ectoderm-derived cell. So how does that exactly occur? I don't know if it's direct, I don't know if it's indirect, but again, another topic for future study. And then it'd be worthwhile to replicate our methods from this study in another animal model devoid of the ability to express p63 in a so-called knockout mice model, and then implant ADSCs, track and evaluate for a return of epithelial cell layering over time that I referred to earlier in this discussion. But at the end of the day, what are we able to walk away with from this study clinically? I feel that what we gathered from this study is that we can complement our understanding of how we explain to our patients that fat rejuvenates the overlying skin, and that the mechanism may extend beyond the paracrine mechanisms that have been discussed in great detail, as I previously mentioned. This does lend additional insight into explaining the so-called stem cell effect to our patients. So I would like to acknowledge SIU, where I trained in plastic surgery, and all of the members of the microsurgical research lab that made this study possible. And I also want to thank the ASJ and the 2014 research panel for selecting this as the research paper of the year in 2014. Thank you.